Good morning. My name is David Aanensen. I'm Director of the Centre for Genomic Pathogen Surveillance, and it's a real pleasure and honour to give this talk really sort of focused on some of the global lessons and approaches for using genomics for pathogen surveillance. I'm going to sort of really focus overall on health system strengthening, really about how we can simplify the use of genomics and digital epidemiology, the additional information that can bring value to genomics for infection control. And then some ways around linking, visualizing and bringing that data to life for local, national and international focus. And then I'll touch on some work that's been done around enhancing national AMR surveillance programs. We're really at this position in time where this technology is really becoming readily available. But some of the key challenges are around how do we integrate genomic information at a very local level to bring value for the patient, but also then at a regional, national, continental and international level. So I'm based at the CGPS. You'll have heard and interacted with members of the team over the last couple of days, Sophia and Julio, Natasha and Nabil. And we're really an organisation that has three major foci. One is around research and genomics, really understanding large-scale pathogen data sets. Implementation, how do we develop technical support packages for the use of genomics in different settings, lab, bioinformatics, etc. And then we have a real focus on engineering. How do we develop tools and methodologies that really support the translation of the research and the understanding that comes out of large genomic data sets through implementation for public health value. So some real sort of simple background, and you will all be more than familiar with these concepts. So within infectious disease epidemiology, one of the real key things that we try and do is identify around pathogens where those pathogens are, what they are, when they cause disease, and who or the population that they cause disease in. And can we distinguish related cases that might be parts of an outbreak or a particular variant and clone from those that are unrelated or not causing particular disease. And we really try and focus this generally, broadly as all of us around, can we identify transmission and look at the spread of pathogens within institutions, healthcare institutions, hospitals, within the environment, food production plants, farms, etc. And can we bring that information together so that we can understand spread on a broader geographic scale, nationally, internationally, and ultimately globally. And with genomics, you take a bunch of samples, you run those samples through your genome sequencing machine, and then one of our ways of getting to this operational unit for genomics is to depict the relationships between samples as a tree. So how similar is sample A to sample B, and what does that tell us about the spread or the epidemiology of the bug under investigation? And because we have genomes, genome sequencing gives us the entire genomic complement. We can also then look into that genome for particular genes or markers that might give us some indication that the sample we've taken is of a greater risk. So if we think about antimicrobial resistance, for example, looking into the genome for genes or SNPs or mechanisms of resistance that might give us some indication on strategies for control. And really there's a vast amount of information that can be brought together on both sides of this figure, from the epidemiological and clinical information on the left-hand side, geography and time and space, and also then the vast information that's generated from the genomics. And one of the key foci is how do we distill that information to deliver rapidly situational awareness that can enable someone to take a decision, an intervention decision, and that individual could be sat at any of these scales at a local level, national level. In the UK we have the UK Health Security Agency, it's our national public health institution, upwards to organisations that are on a continental level like in Europe ECDC, or in Africa the Africa CDC, and then ultimately globally, can we bring information together rapidly to deliver situational awareness at all of these scales? And of course we know that genomics enables us to really sort of plan and develop better interventions, better diagnostics. Can we use diagnostics to identify particular clones or variants? And genomic information has been used to develop vaccines, and there's an increasing interest in bacterial vaccines, as all of you will be well aware around the work within Klebsiella. But also genomics enables us to really go for precision targeting around therapeutics and the identification of outbreaks and transmissions. So there's various sets of information, simple axes of information, sets of information that can be used as public health units to allow us to describe the dynamics of the pathogens that we're looking at. So if we think about COVID was a virus, SARS-CoV-2, and one of the nomenclatures that was developed were the pangolineages, B117, that then were translated by the WHO into the nomenclatures that we're all familiar with, alpha, beta, omicron, etc., which is the variant, and then our mutations to keep track of whether those variants are becoming more transmissible, more virulent, mutations of note, risk markers. And these sets of information are appropriate and parallel within bacterial and fungal pathogens. So within bacteria, for antimicrobial resistance, some of our variant and clone designations are things like MLST, CG-MLST. It gives us those public health units that if I'm talking about a particular type, somebody else can understand what they mean. So it allows us to have this public health unit to be able to then overlay information on top. And within a paradigm such as AMR, our risk markers would be the determinants, the genes and the SNPs, or for vaccine development, serotypes, etc. So this simple set of information, when it's combined with geography and time, really allows us to have units that we can monitor where they are in space and time and how they're spreading, to be able to develop better ways and approaches to control. Now again, back to some sort of pragmatics. This all relies on information coming together rapidly and in a time scale that enables these interventions to come together. So we spend a lot of time thinking about these levels of data architecture. So if we think of any public health institution or any location, there are systems present within those agencies or locations that contain data. And that data, when combined in the right way, can be used to develop and address a set of questions that we might want to answer from that data, that then gives us some information to be able to make change. So the information being the landscape of questions, is there a variant that's increasing in space and time, and what disease is it causing, and how severe is that disease? Knowing that information and bringing it together really allows us to then think about making changes and what the impact of those changes might be. So we spend quite a bunch of time within these arrows trying to think about standard processes that may be able to be addressed, that can help speed up or delineate or really support the development of information flow up and down this chain. So let's step back a little bit and think about AMR. So we do have globally an approach that's developed by the WHO for collating information about phenotypic resistance. The Global Antimicrobial Resistance and Use Surveillance System that's focused by the WHO is a global effort to try and collate phenotypic information from member states into a global entity so that we can understand trends in resistance over space and time. And this consists of this kind of global entity that national entities feed in. So there's an information flow from national public health agencies through the development of national action plans, national implementation plans, etc., that's focused on supporting both regional and national endeavours, but also then feeding information up to get a broader global picture. But if we then scale down into a national level, some of the approaches are within AMR national coordinating centres that then hook into a set of surveillance sites across a country. Could be hospitals, could be other entities that then really touch on the patient. So the patient presents at a surveillance site, that information is then fed upwards to a national coordinating centre, which then feeds upwards into national control, but then also ultimately up to that kind of global level. And then we could go down another level. So this is, on a continent, you can imagine those continental public health agencies collating information from the nodes, the countries within each of the continental level, Within continents, there are national continental agencies, for example, the Africa CDC, which is focused on collating information from member states across a continent. We can go down further into a particular country. Here's Nigeria. The Nigeria CDC is that top level node that has sentinel sites feeding into a national view. And then you can go down into state level. This is Ibadan state, and Ibadan has an entity that collates information from hospitals. So this is this kind of fractal landscape which sits upon our ability to go from patients all the way upwards to local, national and international streams of data. Now what happens at a national entity? So, you know, a national public health institute, with bioinformatics we have samples that come into a lab, genome sequencing is undertaken, bioinformatics is undertaken, and decisions are made within this process. You know, information is passed to the lab director, who then passes that information potentially to the Ministry of Health or the director of the national public health agencies. But these are kind of templates within which we can try and understand how data flows and try and kind of work towards processes that improve that data flow to maximize the value of information for local and national utility. And some of the things that we've sort of focus on are the development of standard systems and processes that really focus on both sides of that spectrum, being able to enable rapid data collection, visualization and linkage at a local level, but then also upwards to how do you maximize our global genomic information to deliver value at a very local level and national and international summaries. So I think you've seen some of the systems that we've produced over the last couple of days. MicroReact, which really sort of rapidly helps bring together data variables, time, space, and any other variable that you might want to map on a phylogeny or a tree that indicates the similarity of a set of genome sequences to each other, that can then help build our hypothesis and understand what the dynamics of the data set that we're looking at is telling us. Now, these trees are very valuable and very useful for small-scale data sets, but if we think of during the pandemic, we now have over 30 million SARS-CoV-2 genome sequences, and of course these trees get really big fairly quickly, and these trees become relatively useless in understanding at a very, very small scale, which is where the development of these kind of broader nomenclatures that define variants or clones or clusters that are more similar to each other than they are to other clusters become useful public health entities for us to talk about international and national spread. So the nomenclature that the WHO developed that gives a friendly way of describing a clone rather than the B111, etc., alpha, beta, etc., becomes our unit of public health. And that then allows us to move away from sort of broad large trees towards visualizations such as this. This is a time scale on the x-axis, and then the y-axis contains distribution of variants within a particular location. This is actually a snapshot from the UK's sequencing efforts across the pandemic, where you can see at the beginning of the pandemic there were multiple variants circulating, and then there were sweeps of particular variants. This is alpha and beta. This is actually an older figure, but this red bar here are the first cases of Omicron in the UK, and you know what happens next, right? There's a sweep of Omicron, but it's different ways of visualizing the information that gives us much more utility to be able to describe what's going on. And these systems are used broadly, you know, being able to place samples, sequences, trees, etc., on different scales to be able to give situational awareness at national levels. Now back a little bit towards the local challenges. So it's quite clear that if you visit any institution, and if you think about the institutions that you're all from, there are vastly different data systems in place. And those data systems again contain the information that enables questions to be answered. And there are common questions that we're all trying to ask in any country in the world, common epidemiological and policy questions that genomics can enhance But even without genomics, there are differences in systems that make it much more difficult to bring information together. And we need to work upon ways or systems that can enable better data linkage and information flow. And we need both bottom-up and top-down solutions, because we're never going to have systems that are present in every organization in the world, but we need to be able to bring different IT and data systems together in a way that allows us to answer these common questions. So one of the tools that we've developed is something called data-flo, which is really focused on local linkage of data systems that might not in a standard fashion be linked to bring information out and bring it together to address those questions. So very, very simply, here's a picture of a data flow. So let's imagine an organization where there are data stored in various different systems. So you have an Excel file that contains some data. You have a text file that is sat on a server somewhere that contains some data. You have trees and you have databases. What this allows us to do is to define what our inputs are, so link to those input systems, carry out a series of transformations, so to bring the information together to address our questions, and then put that information either into another system or to a visualization to be able to standardize in this middle section a common coded epidemiological problem. And these kind of approaches have been used in public health agencies to really rapidly bring together data from different systems and then to be able to visualize the outputs with respect to common questions. So this is a snapshot from Public Health Scotland on the right, which is the distribution of SARS-CoV-2 variants that then sits upon a set of different systems. But of course, you can then transport these data flows into a different organization and tailor the data sources to the makeup and the architecture of that particular institution. And what we see is actually you can very, very rapidly approach bringing data from different systems together to address different questions, such as very, very simply, if somebody is in the lab one day merging two Excel files together and they're doing that on a regular basis, that takes a bunch of time, right? But if you can code a very simple data flow that pulls in those Excel files rapidly and merges them, you can automate some of these processes without replacing your base level data systems such as Excel. And we see these approaches being used for all sorts of different paradigms. So back to genomics. So you've utilized Pathogen watch. So as a general kind of principle, what are some of the things that one needs to do at a local level with a genome to get some valuable information from it? So on a single genome level, what we want to do is pull out any kind of markers or anything that's in that genome that gives us some value for decision making, such as is there a gene present that we think may well inform on a particular phenotype? Is there a serotype present, a KNO antigen type that may well indicate that if you have, for example, a vaccine, that it will or will not work? So very rapidly pulling out of a genome a report that gives you some of these yes-no answers. But then, of course, combining that information together enables additional questions to be understood, such as, is there an outbreak going on? Is the genome that I've just looked at similar to genomes that are very local or elsewhere? And that's really focused on how do you cluster those genomes together and rapidly report that information for useful decision making. And then ultimately we can bring that information together on a much more broader scale to be able to monitor the emergence of new clones or variants of risk. And that information should be able to be used for developing better interventions or monitoring the emergence and spread on a much more broader scale. And I think you've touched upon the use of Pathogen watch in this area, but it's all of the work that you're doing, all of that bringing the genomics together to be able to then address much more broader questions, but also being able to inform at a very local and regional level. So I'm just going to talk a little bit now about some of the work that we've been undertaking within the realm of antimicrobial resistance. So we host the WHO Collaborating Centre on Genomic Surveillance of AMR, and it's really been built upon work within a specific project, which is an NIHR Global Health Research Unit on genomic surveillance of AMR. And this has been a project we've been involved in for the last 5 years, and it's a collective partnership. We work across 4 countries, so within Colombia, AgriSavia is a One Health agency. It's a public-private partnership that feeds together information from human, animal and agricultural health. Within Nigeria, Ruka, who's the co-PI on this grant, has really developed the National Action Plan for AMR and works very closely with the Nigerian CDC, really about building sentinel infrastructure and national infrastructure for antimicrobial resistance surveillance. In India, Ravi runs the KIMS teaching hospital and is networked into a whole series of sites across India. And in Philippines, Celia and Sonia Shah, who is now the lead in the Philippines, is based in the Department of Health. And in the RITM, they have a very long-standing AMR surveillance program, which I'll touch upon a little bit. Part of this project was about how do you enable and place genomics on top of routine antimicrobial resistance surveillance to try and increase capacity for national AMR surveillance programs. And then we within the UK focus on some of the pieces that I've mentioned before around data-flo and supporting technical support for bioinformatics and interpretation. So I'll touch a little bit upon one of the locations and the situation in the Philippines. So in the Philippines, they have a long-standing 30-year AMR surveillance program focused on phenotypic information from sentinel hospitals and WHO priority pathogens. And what they do is on an annual basis, they report on bug-drug combinations and rates of resistance across the country. And that's done through a series of sentinel sites. So there are sentinel hospitals around the country who have standard processes for phenotyping samples that come from patients. And this is the kind of information that comes out. So you have resistance profiles that indicate phenotypic resistance to a series of antibiotics. And that information is used to collate and understand trends in resistance over time. And that information can be used for surveillance stakeholders, either at the local hospital level, the national level, prescribing habits, etc. And then ultimately, that's the base level information that's fed into the GLASS program at the WHO. So this is exactly this is a figure from one of the national annual reports, which indicates changes in resistance, proportion resistance over time for bug-drug combinations, in this case 3 antibiotics to Neisseria gonorrhea. Now you can see that the trends are the lines go up or the lines go down, but actually what does this line increasing here mean? Is that a clone that has emerged in the Philippines? Is it a clone that has come from another location? And these are the kind of questions, and if you have that greater precision, there's better opportunities to implement infection control. So one of the things we wanted to do is to layer genomics on and try and understand what these kind of lines, what these lines mean. So within the Philippines and across the GHRU, technical support packages were developed around placing a genome sequencer into a lab. What's the standardized SOPs across the 4 countries that would enable DNA extraction to be done in standardized fashion, AST testing in standardized fashion? What's the kind of standard bioinformatics processes that need to be used to pull out information from any genomes that are developed? And then data-flo, which is linking the information to other data sources. So this was a manuscript that was published, which was around the process for standardizing implemented genomics in each of the locations. So back to what additional information can now be sought from adding genomics on top. So now in the Philippines, they're able to describe samples with respect to what the strain is. So, you know, the sequence type, what's the public health nomenclature, and also what's the mechanism of resistance. Can genes or mechanisms be identified that give a clue as to what is causing the resistance, and whether that resistance mechanism is carried on the chromosome or on a plasmid. So really fine-scaled granular information that can be much better used to be able to inform potentially public health information and implications at those local, national and international levels. And one of the things that the Philippines have done is that they've implemented a combined approach called Spot and Stop, which really utilizes all the phenotypic profiles from all of the sentinel sites and then clusters and looks for patterns that look like they are outbreaks. Those outbreaks are then confirmed with whole genome sequencing. So actually, if you have an outbreak, if you have a cluster of phenotypes, it's not necessarily a particular outbreak. It could just be that you have multiple mechanisms. However, within this manuscript, there's a description of identifying, through phenotypic cluster detection and confirmatory whole genome sequencing, the presence of long-term outbreaks of Klebsiella pneumoniae in neonatal intensive care units, which then led to infection control interventions that stop those outbreaks happening. So it's a real, all the way back to the hospital level implications of what can be done with the data. So again, being able to use heightened information and precision from genomics to address additional pieces. And these are the kind of things that we see emerging, the ability to spot and develop rapid diagnostics, and ideally to be able to use this information to inform the development of vaccines. And I think that's a lot of interest to everybody in the room here, but also then being able to monitor the impact of those vaccines. If you vaccinate against a set of serotypes, are you paving the way for more pernicious strains to come into a population? And genomics enables us really to monitor that in a way that's unprecedented. So just to touch on bringing the information together a little bit more broadly. So genome sequences that have been generated, that the public genome archives are a rich resource of information where anyone in the world who's sequenced information can place their data in as an archive to be able to actually then open up avenues of research, but also to be able to inform information going forward. So one of the challenges with the archival information is that it's unstructured, minimal metadata, vast amounts of efforts to get increasingly better metadata, but it's really an unstructured archive. So one of the things we wanted to do was to, with a lens on AMR, focus on, can we look at and develop something that really gives us some better situational awareness of all the genomic information that exists. So we have a very nice framing piece of work from the WHO. Now this is actually the previous list of priorities. There is an updated version, but clearly I haven't updated my slide yet, so apologies. But these bug drugs have been identified by the WHO as global priorities for the development of new antibiotics. So this gives us a real frame to be able to look into the archives to try and understand what is the information telling us. So the team undertook a piece of work where any genome that's in the archives that has both time and place was pulled out, quality controlled at a standardized fashion to try and only utilize high quality information to then deliver a set of genomes that are usable within a public health description, and then undertaking the identification of resistance determinants in those genomes, but also then looking at that other piece of axis of information, which is what's the way of defining a clone. So can you define a variant clone and also then piece together the resistance data? So we wanted to be able to deliver this information in real time as data comes into these archives in a format that could be used for local and global policy. So AMR Watch is an initiative that allows you to go and explore the current global landscape of bacterial genomes that are within that priority pathogen list. So just back to one of the visualizations I mentioned about trees. So this way of viewing the distribution of variants over time is also applicable within our sphere of antimicrobial resistance. So let's see, this is a cartoon mock-up. This figure on the left shows over time the distribution of a variant or a clone. And then on the right hand side, these bubble plots indicate for a particular clone the frequency. The size of the bubble represents the frequencies. These two visualizations in tandem can give us quite a lot of information. So if you visit AMR Watch, we wanted to present this information in a simple fashion. You can choose any of the priority pathogens from a drop-down list, and they're bracketed by the WHO groupings. And this little tick here essentially represents that what you're looking at is an always-on, up-to-date view. So any genomes that fall into the archives are automatically pulled through QC to search for the genotype and the mechanisms, and then presented here in context. So this then, we've chosen Klebsiella pneumoniae. This then gives us a global view of the number of genomes that are present, and also their geographic distribution, their variant distribution, and then this is the plot of the variants over time, as well as the proportion of what you're looking at, the data that you're looking at, that contains one of the resistance mechanisms, in this case for carbapenems and cephalosporins. But of course this needs to be interactive and updated, so you can click through down to, for example, looking at a continent level, and you can see here that the light blue areas show that there are some genomes in those archives, and the pink represents that there's no data available, and that really we want this to be able to over time show the increase in data on a global level. And then you can go down and click on a country, for example. So this then for South Africa shows the variant distribution, what are the variants that are in that genomic data over time, and what's the frequency of those that are resistant to carbapenems or cephalosporins. Now, what this does highlight, there's only 364 genomes in those public archives that are of a particular quality that also have the metadata of time and place. Now, one of the reasons why we wanted to push this out was so that we can start thinking about how do we improve our situational awareness on national and international levels. If you think about SARS, 30 million genomes is a pretty reasonable sort of size data set, but we have roughly about 500,000, sometimes a little bit more than 500,000 usable bacterial genomes in this space. So there's a real need to move forward. We need vast improvement, both within the ability to represent data within the genomic archives, but also broader scale sampling strategies. And I think, again, the work that you're all doing around both structured surveys but also bringing together samples from across different countries really provides opportunities for countries and locations to exemplify the approach for bringing this information together in a much more defined fashion. To then think about, can we monitor over time the changes in the clonal population to then trigger diagnostic development, vaccine development, therapeutics, and of course the identification of outbreaks and cross-border spread, similar to how I described in the Philippines. And of course, as you get this rich set of information, we can think of all sorts of other pieces that can be built on top. Can we use the genomics to help inform our estimates of burden, bringing phenotypic data to do the phenotype-genotype correlations, etc., etc. So I'm going to finish there. Really, the talk was kind of broad and slightly rambling at times. I apologise for that, but really, we're really focused on health system strengthening. How do we collectively, globally focus on real delivery for public health decision making, both at a very local level, but then also nationally and internationally? And we really need these data processes in the hands of people that understand the questions, that's all of you and everybody else, to be able to build ways of answering your own questions in a much more rapid fashion, with a real focus on bringing information together to identify and monitor the change in our bacterial populations, so that we can understand both how these things are evolving, but also better be positioned to develop and instigate interventions. So I'll just thank everyone, particularly Sophia and Julio are in the room. I'm always inspired to have the opportunity to work with very talented individuals and the rest of our team, and thank you very much for listening.